thank you for the introduction. My name is Juan, and I'm from University of Minnesota. And today I'm going to show some results we have about in vivo transcranial imaging of blood perfusion in rat brain using contrast-enhanced ultrasound. So first motivation, our lab is interested in working on transcranial focused ultrasound-enhanced stem cell therapy for stroke in small rat model. And the DMUA we have is capable of generating localized subtherapeutic thermal and mechanical exposure. So in the publication of Dr. Haratonova from our lab has shown that the DMUA is able to provide the temperature, the real-time temperature feedback for controlled exposure. So all this promising work gives us the feasibility for the stroke treatment. So to achieve the stroke treatment, use the minimum exposure in order not to damage other brain tissue. Blood perfusion is very important for providing feedback for planning and assessment for the treatment. So in this slide, the left figure is a schematic of the DMUA transducer we have. As you could see here, this is a concave transducer and 32 elements aligned here. And so the DMUA is able to give us the focus inside the brain even in the presence of the skull. So the center figure shows the experiment setup, which I will give you more information in the later slide. So this figure is the even blue dye injection result from the blood-brain barrier opening experiment we have in the late September. And as you could see here, we were able to use the thermal effect to open the blood-brain barrier. And all this promising work and give us the feasibility for the treatment. Still, we need to use the blood perfusion to provide the guidance. So the objective of this paper is to investigate the feasibility of imaging the perfusion activities using in vivo low mechanical index transcranial DMUA imaging. So to be specific, we use the synthetic-aperture imaging under the low MI, about 0.05 to 0.11. So the advantages of synthetic aperture is that it transmits single element each time, which could give us the uniform excitation throughout the brain from different angles. And low MI will minimize the nonlinear signals from the tissue. And then we use the third-order Volterra filter to increase, to capture the nonlinear signal and increase the dynamic range for the perfusion imaging. And following that, we use the temporal perfusion index to quantify the perfusion dynamics, which I will talk about more later. So in this movie, we have shown the raw data we collected during one experiment. So this is before and after injecting the contrast agent. For the display purpose, we scaled the data 20 to 255. As you could see here, after the injection, there is a slight enhancement in the center of the brain, but it's limited. And so the lower level is the echogenicity plot of these pixels we are chosen here. And before the injection, as you could see here, the pattern is very parodic. You could see the pulsating and the breathing gasps. But after the injection, the pattern is very unpredictable. And another thing you should notice that echogenicity is enhanced after the injection. So to achieve better perfusion imaging, first we apply the third-order Volterra filter. So Volterra filter is a commonly used nonlinear filter, which could give us the linear and nonlinear components. And in the previous talk, the speaker mentioned about different methods for perfusion imaging, like pulse inversion and subharmonic and harmonic imaging. And the advantages of the Volterra filter is that there's no sacrifice for bandwidth. So the Volterra filter will capture nonlinear interactions at all preserved frequencies. As long as f1 plus f2 equals 2f, we will capture it in the quadratic component. Another advantage is that Volterra filtering could further improve the signal to noise ratio, and also give us the high dynamic range to image the heterogeneous perfusion. So even with nonlinear filtering and the echogenicity from macro vessel is still very subtle, and to better improve the blood perfusion from macro vessels, we propose the temporal perfusion index. So the main idea of the TPI is to reward the variance of each pixel over frame, which is over time, and at the same time penalize the mean of each pixel over time, and with a special moving average filter. So even we use we apply special moving average filter, the variance is computed at pixel-wise basis, so there is no sacrifice for the spatial resolution. And for the result I'm going to show here, the breathing motion effect will be accounted for. So this is our experiment setup. So this is our DMUA transducer, and the rat is laid down in the platform under anesthesia. And here we use the ear bar to suture the hand, try to minimize the motion effect. And we use the Targestar-P from Targestar, California, and the dose is about 60 microliter contrast agent with 140 microliter sterile saline. And at the same time, we monitor the ECG signals and to make sure the animal is safe during the experiment. So the first, here comes the result. So first, I would like to show you the TPI results with different dynamic range. So here we show the linear, quadratic, and the cubic component. So all three components shows the separation before and after injecting the contrast agent, and nonlinear component has better separation compared to the linear component. And another thing we should notice that is Volterra filter extended dynamic range significantly. If you see the linear component is under 100 dB, but if you see the cubic, it's almost toward to 200 dB. So which give us higher dynamic range to track the blood perfusion. And for each component, it has its own dynamic range, and but it's not critical. So for this case, 50, 120, and 170 is determined to be the optimal dynamic range for each component respectively. So so this movie I'm showing here is the TPI result from the linear component. And so the TPI is showing the false color overlay the beamformed image and before and after. This is the TPI plot of 1 pixel over the frame. And the pixel was chosen here because it has the largest TPI value before the injection. And as you can see here, there is a clear separation before and after, and multiple locations were tracked here. And this is a maximum projection of the TPI over the linear beamformed images before and after. And to get a sense of the spatial resolution, here we use a 6 dB bandwidth. And for this specific location, the spatial resolution is about 250 macro in axial, 350 macro in lateral. And so this is the result from quadratic component. And so the pixel is slightly different, but the same standard. It has the largest TPI value before the injection. And as you can see here, quadratic has better separation compared to the linear component. And the image is much cleaner than the linear component, and multiple locations is tracked. So still the maximum projection from the quadratic component, and it has slightly better lateral resolution compared to the linear component. And so this one is from the cubic component, and the dynamic range was 170 dB. And so the result of cubic component is quite consistent with the quadratic component. Still clear separation and multiple locations were chosen inside of the brain. Maximal projection shows that it has similar resolution, the cubic and quadratic. So to get a better sense about two different nonlinear components and also compare the Volterra component with the low data, here we plot them side by side. And as you can see here, linear and quadratic is consistent, and the image is cleaner than the than the low data result. And for this single pixel we are chosen here, the low data is not able to track the perfusion in this location, but for the nonlinear component shows relatively high TPI value, which indicates this is a perfusion location, and quadratic shows slightly better result than cubic component. So in conclusion, we show that the low synthetic aperture transcranial DMUA imaging as a low mechanical index is sensitive to the contrast activity in the right brain in vivo. And Volterra filter component shows higher sensitivity and specificity than the low data. And TPI is able to provide the increased specificity to image the heterogeneous perfusion with high spatial resolution without sacrificing temporal dynamics. And with that, thank you for your attention. I'm happy to take any questions you have.